We stopped our class last day while discussing the variety of the immune cells of our immune system. To recapitulate, the immune system comprises of two arms. One is the innate immunity, which comprises of the cells. There are certain cells in the innate immune system, the major being the mast cells, the dendritic cells, the macrophages, and the NK cells. Eosinophil, basophil, neutrophils. So these are the cells of the innate immune system. Now, there is another arm of the immune system, the adaptive immune system. Adaptive immune system comprises of mainly the lymphocytes. The lymphocytes are again of two types, the B lymphocytes and the T lymphocytes. Usually, whenever there is a pathogenic attack on the body, it is the innate immune system that first takes up the charge of the body's defense, and gradually, after 3 to 4 days, the message is transferred to the cells of the adaptive immune system so that they gradually pick up the message that a pathogen has invaded. The innate immune cells have fought, and now we also need to fight this foreign pathogen. When we are talking about the adaptive immunity, as I said last day, we talked about the T and the B lymphocytes. Among the adaptive immunity, the T and T lymphocytes comprises of the cell-mediated immunity. And when we talk about the B lymphocytes comprises of the humoral immunity. So broadly, once more, immune system, innate immunity, adaptive immunity. Adaptive immunity comprises of the cell-mediated immunity and the humoral immunity. Further, last day when we are talking about the lymphocytes, I had said that the lymphocytes can recognize foreign epitope, foreign molecules or the antigens via certain particular receptors. These receptors are the T cell receptors. These T cell receptors can recognize the foreign antigen when the antigen is complexed with another receptor. For example, this is a T helper cell. This blue color receptor is the CD4 molecule, right? This T lymphocyte will recognize a foreign antigen only if the antigen is complexed with MHC class 2 molecule, which is present on the surface of a APC. So antigen presenting cells often engulf foreign molecules or pathogens. They process the pathogen and finally they present the pathogen via the MHC class 2 molecules or the receptors. It is this foreign particle complexed with MHC class 2 molecule that is recognized by the T cell receptor, and the CD4 molecule recognizes the MHC class 2. On the other hand, when we are talking about the T cytotoxic cells, they can also recognize foreign pathogen, but this is complex with a receptor known as MHC class 1. MHC class 1 receptor is present on all cells of the body. So this is a very vital difference between the two basic subpopulation of the T lymphocyte. So in nutshell, common lymphoid precursor has 3 categories of lymphocytes: NK cell, which functions in innate immunity, B and T lymphocyte, which functions in adaptive immunity. When we are talking about B lymphocyte, we are talking about the humoral immune response of the adaptive immunity. When we are talking about T lymphocyte, we are talking about the cell-mediated immune response of the adaptive immunity. The T lymphocytes can be broadly divided into the T helper cells and the T cytotoxic cells. Now, I just wanted to show you this picture to give you an idea about the MHC molecule. As you can see, this is MHC class 1 molecule. This is how the receptor looks. So we have one alpha chain which is attached to the membrane, and we have a small beta chain. And the foreign particle this is the peptide group, as you can see. So the foreign epitope or the most foreign sequence of the pathogen is processed, and around 8 to 10 amino acids of that pathogen, 8 to 10, remember 8 to 10 amino acids of this pathogen is presented here. If we see an incoming T cell, the TCR or the T cell receptor binds with this MHC class 1 molecule and recognizes the peptide. And the CD8 molecule recognizes that whether it is that this is the MHC class 1 molecule. So CD8 cells interacts with the MHC class 1 molecule. So all the lymphocytes bearing the CD8 receptor can interact with the MHC class 1 molecule. MHC class 1 molecule is present on the surface of all the normal cells of the body. They are usually not always upregulated. They are only upregulated if a normal cell is infected. If a normal cell is infected and if there are abnormal proteins being produced in the normal cell, these abnormal proteins are then broken down, and 8 to 10 amino acids of the most foreign part is then selected and exposed to the surface with the help of MHC class 1 in their peptide group. When you're talking about MHC class 2, it consists of two different chains. It has a transmembrane domain. MHC class 2 is present on the surface of all the antigen presenting cells. So they have exclusivity. All the antigen presenting cells can engulf foreign particle, break down the foreign particle, select around 30 to 18 amino acids sequence, which is most foreign, and present it in this groove. This antigen MHC complex is then recognized by the T cells with the help of the T cell receptor, and the CD4 molecule recognizes the class 2 molecule. So CD4 lymphocytes recognizes peptides or antigens when they are complexed with MHC class 2. So this is the basic difference between the CD4 molecule, which interacts with the antigen presenting cell, CD4 bearing T lymphocytes, and the CD8 bearing T lymphocytes, which interacts with any normal cell of the body which has become abnormal. The CD4 molecule actually helps. It cannot kill. But the CD8 molecule, if it binds to a cell which has now become abnormal, it will finally bring about destruction of the target cell. You people have studied apoptosis. CD8 positive T cytotoxic cells can express the Fas ligand. All normal cells of the body usually have the Fas receptor present in monomeric condition. They're present as monomers in the plasma membrane. Whenever there is this MHC class 1 molecule expressing an aberrant protein, the T cytotoxic cell binds to that target cell. Then it expresses a CD95 ligand, so that the CD95 or the Fas receptors starts forming clusters of 3 on the target cell. Ligand receptor binding occurs, and the target cell is then instructed to die initially by the extrinsic death pathway, followed by the roping in of the mitochondria or the intrinsic death pathway. So cytotoxic T cells can kill a target cell, but that is the CD8 positive T cells are killers. But on the contrary, the CD4 positive lymphocytes are helpers. They help in propagating the immune response from the B lymphocytes or from the dendritic cells, that is cells of the innate immunity, to the B or the T cytotoxic cells. So they are mediators. They carry the information from the innate immune system to further down into the adaptive immune system. Just a second. This is a very simplified way of how peptide molecules are presented in case of class 1 MHC complex, and peptide molecules are presented in case of class 2 MHC complex. As I said, in case of class 1 MHC complex, when there is presentation, you can say a virus has entered a cell. So this is a viral infection. So virus has entered a cell. We are having the viral DNA or the RNA. And using the host cell machinery, the viral DNA or the RNA is producing the viral proteins. The viral proteins are not normal proteins of the body. They are aberrant proteins. These aberrant proteins are then expressed on the cell surface by the MHC class 1 molecule. The main reason is that a T cytotoxic cell will see them, will bind with their TCR to this MHC class 1. The CD8 molecule will recognize this, and then this T cytotoxic cell will instruct this viral infected cell to die an apoptotic death. So this is the presentation of viral proteins via MHC class 1 molecule. Now let us look into the MHC class 2 pathway. This occurs specifically in case of antigen presenting cells. So we have a restriction here. MHC class 2 is not present in all cells. Again and again, I'm stating this fact. It is present on the professional and to some extent unprofessional antigen presenting cells. So these antigen presenting cells, these cells are capable of phagocytosis. That is by law. All the antigen presenting cells are capable of phagocytosis. So they are capable of ingesting an antigen and engulfing the antigen. They have formed endosomes. Within this endosomes, lysosome fuse. You all are aware of what happens when lysosome fuse. Lysosomes have a lot of degrading enzymes like proteases, like cathepsins, and so on. These lysosomal enzymes then degrade the foreign pathogen that has been engulfed, and a small antigenic part of the pathogen is then presented via the MHC class 2. When this pathogen is presented by the MHC class 2, they are recognized, again, as I said, by the TCR, and the MHC class 2 is recognized by CD4. So in this case, T helper cells are activated. Once T helper cells are activated, depending on the signals and the cytokines liberated, there is a decision made. So we can move to a T helper 1 category or to a T helper 2 category. If we move towards T helper 2 category, then finally it is the activation of the B lymphocytes. If the T helper cell move towards T helper 1 category, then it is the activation of cytotoxic cell. So you can understand that when a naive, a fresh T helper cell interacts with MHC class 2, there is a decision to be made by the T helper cells whether to enter the helper 1 pathway or the helper 2 pathway. This decision is facilitated by the production of cytokines at that environment. The cytokines helps the T helper cell to decide which pathway it needs to opt, because the pathways are defining. If a helper cell decides I should enter helper 1 pathway, it cannot revert back to helper 2 pathway, and vice versa. If a helper cell decides I want to enter the helper 2 pathway, it cannot revert into the helper 1 pathway. But this decision is very vital, because once this decision is made, and the helper enters the helper 2 pathway, you can understand it finally activates B lymphocyte. So the humoral immune activation is occurring. But if the naive helper cell decides to enter the helper 1 pathway, then it activates the T cytotoxic pathway. That means it is further activating the cell-mediated immunity. So from the earlier slide, we understood that the helper cells interact with MHC class 2 molecules and the cytotoxic cell interacts with the MHC class 1 molecules. If this interaction is successful, then the cells expressing this MHC class 1 molecule will die. If this interaction is successful, then as I said, a decision is to be made whether the helper cells will proceed in the helper 1 pathway or the helper 2 pathway. Let's see another picture. This picture is a beautiful picture. See, depending on what kind of pathogen is actually entering the body, the decision is initially made. So we have pathogens. Pathogens can promote, just a second. So you can see that there are pathogens that to some extent determine which pathway the adaptive immunity should follow. So there are T helper 1 promoting pathogens. There are T helper 2 promoting pathogens. When I talked about innate immunity, I said that one of the important players of the innate immune cells are the dendritic cells. And these dendritic cells can recognize the pathogens by the presence of the molecular patterns on the surface of these pathogens, which are known as PAMs. So these PAMs are recognized by receptors present on the dendritic cell, which are known as pattern recognition receptors. So TLR, toll-like receptor, is nothing but a pattern recognition receptor. So it is this receptor that first identifies the molecular pattern and gets an idea that whether this molecular pattern will start a helper 1 pathway or a helper 2 pathway. So accordingly, the signaling cascade starts. Now the question is that you might be a bit confused by the fact that how can a receptor decide between a helper 1 PAMP and a helper 2 PAMP? There are around 11 to 12 categories of toll-like receptors. And each receptor is specific for a particular PAMP. So activation of TLR4, for example, that is a toll-like receptor 4, that can recognize viral PAMs. So if there is a virus entering, so it will be able to recognize the viral PAM and that automatically will proceed for the helper 1 pathway. But again, if it's a parasite that has entered, then there is a separate category of toll-like receptor that can recognize the helminth PAMP. And accordingly, that receptor signaling will start the helper 2 pathway. So it's not one receptor. There are multiple receptors and each receptor has a defined signaling pathway and each receptor can recognize a defined pathogen associated molecular pattern. So that is how the recognition is done. It is not one receptor which is distinguishing between both TH1 PAMP and TH2 PAMP. So we have separate receptors. Some receptors for TH1 PAMP and some receptors for TH2 PAMP. So it depends on that whether the pathogen is binding to the pattern recognition receptors associated with the TH1 PAMP or the pattern recognition receptor associated with the TH2 PAMP. This binding actually decides which pathway, which signaling will start. So now, coming back to the story, so when there is a TH1 promoting pathogen product, so we have a toll-like receptor which is capable of recognizing the TH1 PAM present on the dendritic cell. So the dendritic cell is activated And one of the activation cytokines the dendritic cell produces is IL-12. In presence of IL-12 and another activating cytokine, you can see IL-27, you see a naive, this is a naive CD4 positive. This is a CD4 positive cell. So this is a naive CD4 positive T helper cell. Hence it is written as T helper 0. The differentiation has not yet occurred. This naive CD4 positive T helper cells interacts with this dendritic cell. Upon interacting, as I said, a decision is to be made. And the decision is further favored by these cytokines present in the microenvironment. If these cytokines are constantly being present in the microenvironment, the decision is that this will differentiate into the T helper 1 category. So it will produce, these cytokines will help in expression of the master transcription regulator of the T helper 1 cell. And this T helper 1 cell in turn will produce IFN gamma, in turn will produce IFN gamma. IFN gamma will then activate the T cytotoxic cell, or we have moved into cell-mediated immunity. So that is one pathway. But just a second, let me remove. But if it is a helper 2 promoting pathogen product, this has interacted with the pattern recognition receptors that can identify them. And in the vicinity, these kind of cytokines are being liberated. In this microenvironment, the T helper 0 cells or the CD4 positive naive cells differentiate into the helper 2 category. The moment they differentiate into the helper 2 category, they start liberating a host of cytokines. These cytokines are very specific for activation of B lymphocytes. So thus in turn, B lymphocytes are activated and the humoral immunity takes on. So you can see this decision here becomes very vital for the immune system to decide whether I move towards cell-mediated immunity or whether I move towards humoral immunity. Whenever there is an extracellular pathogen, a pathogen that has entered the blood, preferably the humoral immunity is preferred because the ultimate product of the humoral immunity is antibodies. You are activating B lymphocytes. B lymphocytes will produce neutralizing antibodies that will neutralize the antigens produced from the invading pathogen. But if there is an intracellular infection, for example, a virus entering a cell, there's an intracellular infection. Virus cannot be recognized in blood. Unless and until it enters a cell, we cannot say infection has occurred. So there's an intracellular infection. In those cases, whenever there's an intracellular infection, in those cases mostly, the T helper cells are instructed to enter the T helper 1 pathway so that finally the cytotoxic or the killer cells are activated and they in turn can kill all the infected cells bearing the viral load. So these were the two basic divisions of the T helper cells. For our undergraduates, we restrict to this, but I would like to show you that a naive CD4 positive T cell can have so many varieties depending on what cytokine is being produced. So this decision is very vital. So as you can see, these are the polarizing cytokines. When we have IL-2 TGF-beta, it is a regulatory cell. When we have IL-4, we talked about IL-4 just now, it is a helper 2 cell which induces allergic antihelminthic response and it induces humoral immunity. When we have the production of IL-12, IFN gamma, we are having a T helper 1 cell which is inducing cell-mediated immunity. You can understand that whenever these cytokines are the stimulators, the cytokines is actually telling that naive CD4 positive T helper cell to understand which subpopulation it should become. And you can see whenever we are having this cytokine stimulation, you can see there is this master transcriptional regulator. So we are having a defined regulator for each cell population. So, for example, for the T helper cell, the transcriptional regulator is GATA3, fixed for T helper 2. That is not activated in the other subpopulations. Similarly, when you're talking about the helper 1 cell, we have a fixed master transcriptional regulator, that is T beta. So it is the cytokine signals which actually control which subpopulation the CD4 positive T lymphocyte should be, because they form an important category of cells which sits at the interface of the adaptive and the innate immunity. Messages, signals from the innate immunity move to the CD4 positive lymphocytes, and based on what subpopulation of CD4 positive lymphocytes we are getting, accordingly, that branch of adaptive immune system will be activated. So for your undergraduates, let's restrict our knowledge to this much only. And so this is a summary of all the cells we studied: neutrophil, monocyte, macrophage, dendritic cells, plasmatoid, DC. These are the innate immune cells of our body. Again, we have here T lymphocyte, B lymphocyte, plasma cell. We are having, these are again the innate immune cells of our body. So these 8 cells are controlling the innate immune system of our body. The adaptive immune system of our body are the B lymphocytes and the T lymphocytes. The B lymphocytes, when activated, become the plasma cells. So from next day, we'll start the organs.